Good morning, everyone. Till last lecture, we all have discussed about the structure of enzyme, then nomenclature of the enzyme, and today we will be talking about the chemical nature of the enzyme. In the chemical nature, we had already discussed about the absolute specificity, group specificity, linkage specificity, and substrate specificity. Today, we will be starting from the structure of enzyme points onwards. So we had already discussed in earlier lectures that the enzymes are made up of the globular protein. So what is the what is the structure of the globular protein? Usually this globular protein are having the 3D structure. So when we talk about the protein, what are the key components of the protein? Proteins are made up of the amino acids. So same way, the enzymes, as they are made up of the proteins only except few. So these enzymes also possesses the amino acid sequences. Now amino acid sequences having one C terminus, that C terminus is the carboxyl terminus, and when we talk about the N terminus, that N terminus is the amino acid terminus. So same enzyme की case में भी enzymes are the globular protein. Hence, it has the three-dimensional structure. It possesses the amino acid sequence. So likewise, they are having the C terminus and the N terminus. When we talk about the sequence of amino acid, right? Each protein is having the different type of the sequence. So this sequence will be determined with the specific genes. We all have learned transcription and translation, right? We know that the gene decides the sequence. So each and every this enzymes also have the different type of the genes in our body. So this particular genes decides the sequence of the amino acid. When we talk about the other protein having primary and secondary structure and so on. So what about enzymes? Yes, enzymes also do have the primary structure, secondary structure, tertiary structure, then thereafter the quaternary structure. Means it is a protein only, but having capability of the catalysis. Understood? So these particular structures are held together in the position with intra or intermolecular bond like proteins only. The finally offered structure, which is a 3D shape and having the catalytic activity, is designed in such a way that all the catalytic group amino acid, they come in a particular site which is called as the active site. We will talk in detail about active site in the next slide. When we talk about the catalytic activity of the enzyme, so this particular catalytic activity depends on the integrity of the native protein structure, how it integrates. If this particular structure, 3D structure, will distorted, this particular enzymes will not be able to work. Understood? Like other protein, enzyme can also be precipitated with the alcohol. We all have the practical, right? Protein precipitation in our SY. We all have seen that for the purpose of the precipitation of the protein, we were using the alcohol. Same way, if you will heat the protein, you will use a strong acid, you will use the alkali and other reagent like TCA, nitrogen sulfide, etc. You can precipitate it. Now, molecular weight. When we talk about the molecular weight of enzyme, so it varies from enzyme to enzyme. It can be as small as 1200 kilodalton to 10 lakh kilodalton. Can you imagine? In few cases, the enzyme can have the molecular weight more than 10,000 kilodalton also. So depends on the molecular weight, their size can be varied. Right now, next thing, as I told you that the enzymes are the globular protein only, but they may possess the single amino acid chain or matlab monomer, and it can be the polymer. So for that particular reason, the active site varies. Now we will have a discussion on the active site of the enzyme. Now active site, it is active site and substrate. Huh? Now, when we talk about the main power of enzyme, for now, what is the important function of the enzyme? The most important function of the enzyme is to accelerate the rate of reaction. That means it lowers down the activation energy. Now, I told you in the last lecture when we were discussing about the activation energy, that activation energy is the energy which is required to convert the substrate into the product, right? So what is the important function of the enzyme to lower down the activation energy? So when we are talking about how all this thing is happening, so all this is determined by the complex structure of a pocket like site which is present into the it can be depends anywhere, not necessary. This is a schematic diagram, students. It can be present into the middle also. So this site is having a pocket like structure, and this particular site is very specific or highly designed as per the substrate. Understood? So this particular site is known as the active site. It provides proper catalytic group to facilitate the specific interaction with substrate and itself. Reaction which are taken place, whatever means, if enzyme is related, suppose it is lactate enzyme. Okay, so what, how lactase enzyme works? It works on the lactate and convert it into the glucose and galactose, right? So now this particular enzyme will bind to the substrate, that is the lactate, from the active site only. You can see here this schematic diagram which is showing the active site of the enzyme. You can see it is ideal with the substrate, isn't it? So it provides the proper catalytic groups to facilitate the specific interaction with the substrate and itself. Now see, this is the most important point, student, to remember that this active site varies as per the substrate, because this particular site, it can be made up of the 3 to 12 amino acid only. So this particular site will have the catalytic activity amino acid, and these amino acids will be very specific in type of reaction they are carrying out. Now see, in the last lecture we all have already discussed about the various enzyme reactions which are taking place by the enzyme. So whichever category or whichever class enzyme is there, as per their class, they are having the catalytic group present in their active site. Now reactions, all the reactions are taking place into the active site only. This point to be noted. Now what about the other huge structure of the enzyme? The rest portion of the enzyme is just a scaffolding. Only and only active site is the important side. Now it does not mean that the other part is removed, only active site is taken, and then thereafter the enzyme will function. Absolutely not. If the enzyme structure is distorted, the enzymes cannot work. Now, after binding substrate with this particular site, it get converted into the product. The shape, the chemical, and the electrical property of the substrate makes the enzyme to bind it. Hence, it prevents binding of other substrate like non-target molecule, because the substrate is compatible with the active site only. Now, when we talk about the active site, so if we will compare the active site area of the enzyme So it is very small area of the total volume of the enzyme. It may have more than one active site sometimes, not necessary that one enzyme one active site, so it is not the case. When we talk about the monomeric enzyme, that means which are made up of the polypeptide chains, this may have only one active site. Now this active site may be like a cleft or it may be like a crevices. Or when we talk about the multimeric enzyme, now multimeric enzyme means which are made up of the many polypeptide chain, more than one polypeptide chain. So active site will lie in between the polypeptide, but in most of the cases, binding of substrate induces the conformational changes. Note down the term conformational changes. Conformational changes means changes which are bringing up after binding of the substrate molecule. So this particular change will occur after, suppose if enzyme is having the X shape, so after coming in contact with the substrate, the shape will change to the Y, which is the suitable shape as per the substrate. I had already discussed that active site may have more about 3 to 12 amino acid. Active site, now what this active site comprised of? What is making it so special to carry out the reaction? So first of all, it is having the special type of the amino acid which is having the functional group. Now this functional group, now we all know about the amino acids, that this amino acid is having the functional different type of the functional group which are making them different from one another, right? So this functional group is having the affinity with the substrate. So this functional group attracts the substrate and lowers down the activation energy for whatever reaction they are carrying out. So this particular active site functional group act as a template for holding substrate molecule in the correct orientation as well as into the position. How they are bind with the, means how enzyme is binding with the substrate. So in between substrate and enzyme's active site, bonds are formed, and these bonds can be the covalent bond, it can be the ionic bond. So certain type of the bond are formed, but as the substrate converts into the product, so this substrate immediately leaves the enzyme and it forms the, it keeps the enzyme again new position, and this enzyme is ready to bind with the another substrate. So this is the total idea. Now what is the reaction? This is a this reaction is enzyme plus substrate, that is E plus S, which gives out, now see all this reaction is the reversible reaction. So E plus S will gives out the ES, then this ES will gives out the EP, and this EP will give E plus P. Let us discuss the reaction. Reaction is enzyme binds with the substrate, so that is E plus S, and this substrate will after binding will form the enzyme substrate complex. Now when the enzyme substrate complex will form perfectly, so this perfect complex will start reacting, means it happens in fraction of second. Okay, so immediately this substrate will be converted into the product, that is EP, means this product will be bound with the enzyme's active site only, and this EP will immediately gives out the E plus P, that means it will be separated from the enzyme, and now enzyme will be empty, enzyme's active site will be ready to bind with the another substrate. So this way one single enzyme can bind to many substrate, but as substrate converts into the product cannot have affinity to bind with the enzyme again. Understood? So this enzyme is readily available for the other substrate. So small quantity enzyme can convert large quantity of the substrate into the product. Understood? So If now what is going to be happen when we talk about the catalysis reaction? So one substrate can be break into the one type of product. Sometimes in the last lecture we all have seen that in some cases enzymes just add some type of the functional group on the substrate, right? Some enzymes can break the substrate in matlab complex substrate into the two simple from polymer to monomer, right? But some cases we all have seen that the enzyme can bind the substrate, like ligases के case में we all have learned. So all the substrate can be break down into the one product, or it can break down to the multiple product. So this is all about the active side of the enzyme. Now we will be discussing about the cofactor and coenzyme. What are the things which are supplementing or helping the enzymes to work in a more and more efficient way? So when we talk about the cofactor and the coenzyme, so sometimes some enzyme require presence of the other compound also. It means only active site is insufficient to performance, and reaction will be taken place from the active site only, but this active site is prevented to bind with the substrate in the absence of the cofactors. These cofactors are of various types, we will be discussing in detail. So this particular cofactor helps in activating the catalytic power of the enzyme. Now this cofactor are the non-protein substances. Note down the term. The cofactors are the non-protein substances. Is the complete complex of enzyme together with its cofactor is called as the holoenzyme. You can see enzyme is just this much. This particular enzyme will be called as the apoenzyme. Now this coenzyme will bind here, and after binding of cofactor, which provides the catalytic side and coenzyme, everything, this complete structure will be called as the holoenzyme, and the protein moiety of holoenzyme will be called, only the protein part is called as the apoenzyme. Apoenzyme is non-functional part, but it is not like that all enzyme requires the cofactor. Not necessary. The cofactor is not necessarily required. Some enzymes can work without the cofactor only. Apoenzymes responsible for enzyme substrate specificity. Now, when we talk about the complete structure, so it, holoenzyme is a complete enzyme which is having the cofactor as well as the apoenzyme as well as the catalytic side. So this is important for the MCQ. Holoenzyme is equals to apoenzyme plus cofactor. I repeat, holoenzyme is equals to apoenzyme plus the cofactor. Neither the apoenzyme nor the cofactor is catalytic active when they are alone. Until and unless they will not bind, they will not form the holoenzyme, they can't work. They become catalytically active only and only when they are combined. So overall, three kind of cofactors are there. They are called as the coenzyme, prosthetic group, and activators. But whichever cofactors are in this particular example, I have shown you the coenzyme. The point to be remembered here is all the cofactors, they are non-protein substances. Now, first we will be talking about the coenzyme. Name only suggesting that it is having relation with the enzyme, right? So these are the non-protein organic substances. Note down the term organic substances. No, coenzymes are non-protein organic substances. These particular coenzymes are very essential for the efficient functioning of some enzyme. But the point to be remembered here is they do not themselves bind to the enzyme. Coenzymes form the transient association with the apoenzyme. Understood? Then thereafter, during the catalytic reaction, they will remain with the enzyme. It is not like that if they are the coenzyme, they will be permanently bound to the enzyme. When the reaction occurs, so that particular time they will bind with the enzyme transiently, and thereafter they will, as the substrate detaches, they also detach from the enzyme. Coenzyme undergoes into the changes to compensate for the transformation in the substrate. So they make the enzymes in a perfect position to bind with the substrate. Here you can see that they have bound, and this conformational changes they are bringing. This is called as a conformational change. As the substrate is ready to bind, so this will bring the change into that. Now, what could be the coenzyme? What are the example of the coenzyme? So it can be coenzyme A, nicotinamide dinucleotide, that is your NAD plus. Then NAD plus act as a coenzyme to the dehydrogenases by acting as hydrogen acceptor in the Krebs cycle. Some of the enzyme usually function as the transient carrier of the atoms or group. Now, when we talk about the other examples, so most of the coenzymes they are derived from the vitamins. NAD plus, NADP plus are derived from the niacin. When we talk about the fat, that is the flavin adenine dinucleotide, it is derived from the riboflavin, that is your vitamin B2. Then, when we talk about the other cofactors, so that usually these particular vitamins are the large group of the substrate. That's why it is always suggested that take vitamins in appropriate amounts. So because this all most of the vitamins they are the coenzymes for activity of the various enzymes, and when we take them into the appropriate amount, so it helps in the metabolism in a proper way. Functional role of coenzyme is to act as a transporter of the chemical group from one reactant to another reactant. So note this particular point. Functional role of coenzyme is to act as a transporter of chemical group from one reactant to another reactant. Okay. So what do you think? Which type of enzyme will possess such type of the coenzyme? Transferase, right? Because the functional role of the transferase is to transfer the one functional group from the one enzyme to the another enzyme. There are many coenzymes, they have their precursors, and their deficiency causes the disease. Examples are biocytin coenzyme, it is having the precursor biotin, and its deficiency causes the dermatitis. Then coenzyme A, pantothenic acid, which also called as the dermatitis. Then FAD-FMN, it is having the precursor riboflavin, that is your vitamin B2. And if its deficiency is there in the body, it can cause the growth retardation. Then your NAD plus, NADP plus. Now NAD, NADP, you may remember your Krebs cycle and all. So this having the niacin, that is your vitamin B6 as a precursor, and its deficiency can cause the pellagra. Then thymine pyrophosphate, that is having the precursor as thymine, that is your vitamin B1. Its deficiency can cause the beriberi. Then tetrahydrofolate coenzyme is having precursor folic acid, and its deficiency cause the anemia. Cobalamin, having the cobalamin as a precursor, that is vitamin B12, and its deficiency can cause the pernicious anemia. So these are the all cofactors, sorry, coenzymes which are having the precursor with them, and its deficiency can cause the diseases. Now, see, you might have seen that when we are having certain reaction or certain metabolism gaps, if a person is having the iron deficiency or having the low HB, if low HB, that means the iron deficiency is there. So directly iron is not given as a supplement. The person is given as the folic acid. Why? Because this folic acid is important for carrying out that particular reaction. Understood? So this is about the first type of the cofactor. Now second cofactor is the prosthetic group. Prosthetic groups are the metal ions which are bound. Now see, this metal ion bounds very tightly to the enzyme through the covalent bonding. In the biological system, the strongest bond is the covalent bond. So this prosthetic group are iron metal ions, and these metal ions are bound very tightly to the enzyme. Very tightly means having the covalent bonds. When we talk about the molecular weight, so this is having a very low molecular weight. Heme example is heme molecule in the hemoglobin. Now you cannot extract it, right? It is permanently bound. It is not like your coenzyme that they will come, bind and go. Heme is an iron containing prosthetic group. It may function as the electron carrier and oxygen carrier in the hemoglobin. Now see, in the case of the coenzyme, what we all have learned? We have learned that they transfer the functional group. Now here, what is the function here? The function is that they act as the electron carrier as well as the oxygen carrier. Understood? Heme group also found in the catalase and peroxidase, which catalyzes the decomposition of the hydrogen peroxide to the water. There are many other examples. Some cases when we are learning about the other photosynthesis and all, so that time you will learn that many other enzymes are there. They are having the copper and other type of the metal ions as a prosthetic group. Then next is the activators. Now these activators are also the metal ions. These are inorganic monovalent or divalent cations. Okay, it could be potassium, nitrogen, ferrous. This may bind to the enzyme loosely or firmly. Now, what is the difference between the prosthetic group and activator? Students, remember this type of things you must always remember by the comparative study. You compare it from the other. When we were talking about the prosthetic group, we have learned that they are firmly bind. Okay, means they have the strong bonding with the enzyme. They were completely attached. Understood? But when we talk about the enzyme activators, activators are again the metal ion. Understood? Now when we were talking about the coenzyme, they were the inorganic molecules like vitamins and all. When we were talking about the prosthetic group and activators, both are the metal ions only. But what is the difference between two? That prosthetic groups are very tightly bound. And when we are talking about this activators, they are not very tightly bound. This may bind to the enzyme loosely or it may bind to the firmly. When we talk about the substrate, so they how their attachment is with the substrate, so they form complex with that particular substrate and help in making the enzyme substrate complex by shaping the enzyme or substrate in the more appropriate shape. So in short, the function of the coactivator to brings the conformational changes into the substrate. Understood? Where example, calcium ions are necessary for the enzyme thrombokinase, which is responsible for the blood clotting. Another example is the salivary amylase, which requires the presence of the chlorine ion for its activation to convert the starch to the maltose. Understood? So this is about the cofactor, and at this note we'll stop. Next lecture we will be talking about enzyme action and kinetics, which is one of the most important topic in this unit.